Today, we're diving into something truly incredible. We're going to watch how a single cell, in just 21 days, builds a complete, living, breathing chick. It is an absolute masterpiece of natural engineering.
Welcome to Silverlight Education.
In this lecture, we're drawing from Unit 15 of the BZYCT 133 course, and let me tell you, it tackles one of the most amazing questions in all of biology. Seriously, how does this happen? How does a single cell know how to build an entire chick completely from scratch? And what controls this whole incredible process? Well, let's peel back the shell on this biological marvel and find out, one step at a time. You know, every big construction project needs a blueprint. And for a chick, that blueprint is the egg. But it's so much more than just a plan or a shell. It's a totally self-sufficient world, perfectly designed to create and nurture new life. The best way to think about an egg is as a complete life support capsule. I mean, everything the embryo could possibly need for its 21-day journey is packed right inside, perfectly engineered for survival. And what's amazing is how every single part has a specific vital job. You have the blastodisc, that tiny white spot, which is literally the spark of life where this whole story kicks off. The yolk, that's the high-energy power bank fueling all the growth. The albumin, what we call egg white, acts as a protective cushion. And then you have the chalazae. Think of these as safety ropes, holding that precious yolk perfectly suspended in the middle. Now, most people just assume the yolk is all fat, right? But that's not the whole story at all. You might be surprised to hear that it's almost 50% water. The rest is this perfect cocktail of fats, proteins, and phospholipids. It's basically a custom-made energy drink for the developing embryo. Okay, so once fertilization happens, the very first stage kicks in, cleavage. This is all about rapid-fire cell division. It's a total numbers game designed to create a massive cellular workforce for the huge construction project that's about to begin. So this happens through a process with a very fancy name, discoidal meroblastic cleavage. Now, it sounds super complicated, but the idea is actually pretty simple. Because that yolk is so huge, the cells can't just divide anywhere they want. So all the action is stuck in that small, flat disc, the blastodisc, right there on the surface. And this division is unbelievably fast. I mean, from 1 cell to 2, 2 to 4, 4 to 8, you get the idea. By the time the hen actually lays the egg, just 24 hours after fertilization, that one single cell has already multiplied into a layer of about 60,000 cells, called the blastoderm. Incredible, right? And right away, this blastoderm splits into 2 distinct teams. In the center, you've got the area pellucida. This is our main character, the group of cells that will actually become the embryo. And surrounding it, you have the area opaca. This is the support group. Their critical job? To process that nutrient-rich yolk and deliver it to our main character. Okay, now we get to what is probably the most dramatic and crucial stage of the whole process, gastrulation. At this point, cells don't just divide. They pack their bags and embark on a great migration, rearranging themselves to lay down the fundamental body plan of the chick. In the center of all this action is something called the primitive streak. The easiest way to picture it is as a cellular highway. It's a groove that forms right on the surface, and it's here that cells literally dive inwards to start forming the different layers of the body. This is what sets up the whole head-to-tail layout of the embryo. This whole migration creates 3 master layers of cells, and each one has a very specific destiny. The ectoderm, on the outside, is going to become the skin and the entire nervous system. The mesoderm, in the middle, is the powerhouse. It forms muscle, bone, and blood. And finally, the endoderm, on the inside, creates the lining of the gut and other internal organs. Every single part of the chick's body starts in one of these 3 layers. So up to this point, our embryo has just been a flat little disc of cells. But now, a process called body folding starts. Folds pop up at the head, the tail, and on the sides, lifting the embryo up and off the yolk. This is the exact moment a simple 2D sheet of cells transforms into a recognizable 3D body. Now, as if building an entire body wasn't enough, the embryo also starts assembling its own personal, dedicated life support systems. These are called the extraembryonic membranes, specialized organs that only work during development inside the egg. Each of these has a critical job. The amnion is a fluid-filled sac, basically a private swimming pool to cushion the embryo from bumps. The yolk sac is its personal lunchbox, connected right to the gut for a steady food supply.
And the allantois is amazing. It's both a waste bin for storing garbage and an oxygen tank, with blood vessels that exchange gases right through the shell.
And that brings us to the grand finale. After 21 days of non-stop, perfectly coordinated development, the chick is completely formed and finally ready to enter the outside world. So let's just quickly look back at this incredible journey. Day 1, the numbers game of cell division begins. By day 2, that great cellular migration is in full swing. By day 3, the very first organs, like the heart and nervous systems, start to take shape. And after weeks of this complex dance, on day 21, the final moment arrives.
So how does it pull off the great escape? Well, first, it pierces the membrane into the airspace inside the egg and takes its very first breath of air. Then, using a tiny, hard point on its beak called an egg tooth, it starts chipping away at the shell. And finally, it breaks free, leaving its now useless life support capsule behind for good.
And this whole incredible process really leaves us with a final, fascinating question. The amniotic egg was a revolutionary invention that allowed animals to finally conquer dry land. So when you think about it, the protective membranes, the built-in food supply, the hard shell, what innovations do you think were the most critical for making this miracle of life outside water possible? It's a pretty fascinating question to think about. We will meet you in the next lecture.